Let's talk about lipid digestion and absorption. So, digestion begins in your small intestine, and biosalts from liver emulsify fats, okay? Meaning biosalts from your liver break down fats. So, pancreatic lipase, what does pancreatic lipase do, right? ACE tells us this is an enzyme. So, this is an enzyme that hydrolyzes triglycerides. Okay, and what we do is we break the triglycerides into one fatty acid and then two glycerol molecules. And okay, next let's go over a mint cell. So what is that? So these transport lipids into intestinal cells, and within the enterocytes, which is just this is just a fancy way of saying intestinal cells, right? So this word just means a cell of the intestinal lining. So when we get into those, we then we re-esterify them into triglycerols, and then these are packaged into something we call catecholamines, which these are just lipid transporters, okay? So you package all your lipids into this catecholamine, and then those enter the lymphatic system, and then from there we go to the blood via your thoracic duct, okay? Awesome. Hopefully that made sense. So lipid metabolism. So how do we metabolize lipids? So we have something called hormone sensitive lipase, and you literally always just see this as HSL. This is in our adipose tissue, and it mobilizes fats during fasting. So this is activated by epinephrine, cortisol, and glucagon. And glycerol then can go into your gluconeogenesis. And then your free fatty acids bind to albumin, which is a plasma protein. Okay. So when you make, I'm going to touch on this again. So when you make glycerol, okay, your glycerol can then be used for gluconeogenesis, okay? So when we metabolize glycerol, we can then turn it into gluconeogenesis. And then we can then make it go into gluconeogenesis, right? We can use that to then make more glucose, right? Again, this is why we say fats are like a storage, right? When we break down fats, we can then make more glucose. And then your free fatty acids will bind to albumin. And then we need to know this is a plasma protein, okay? I don't know if you've taken anatomy 2 yet or whatever, but this is a plasma protein. And as far as, okay, what's the significance or the function? I'm not actually sure. It wasn't in the book, and I haven't seen it on like my flashcards, but I would definitely know this. Okay. Okay, next we're gonna go over lipid transport, and these is really important, and this is from least dense to most dense. Okay, so catecholamines transport dietary lipids to the tissues. VLDLs transport liver synthesized triglycerols. Okay, IDLs are VDLs remaining. So when we take cholesterol out of VDLs, we then get IDLs, and then those become LDLs, or they can be reabsorbed by the liver. And remember, we don't want LDLs, right? LDL is bad. So LDLs then deliver cholesterol to the tissues, okay? And this is why LDLs is considered bad cholesterol, because you don't really want a buildup of cholesterol in your tissues. And now we call HDLs good cholesterol because HDLs pick up excess cholesterol and returns it back to the liver. So this is actually good for you, right? You're going to take the excess cholesterol and then bring it back to the liver, okay? And this is from, I don't know if I said high to least. No, this is from least dense to highest dense, okay? Least to high. And last but not least, adipoproteins. These are protein complexes that mediate lipoprotein binding as well as uptake and enzymatic function. Okay, so we're going to stop there, and then we're going to go ahead and start with 11.4, cholesterol metabolism.